Hi everyone, it's Dr. LeBlanc with Unit 11A Complications of Mendelian Inheritance, Sex Determination and Sex Linkage. Our learning goals are to understand that male and female sex determination can have genetic and environmental bases, to understand what heterogametic and homogametic sex determination is, to understand that plants can be either monoecious or dioecious, to understand the concept of dosage compensation, to understand the mechanism of X inactivation in mammals, and to understand sex linkage inheritance patterns, and to learn how to recognize them in a pedigree. A lot of animals on planet Earth are sexually dimorphic. There are two sexes, a male and a female. Here's a male cardinal and here's a female cardinal. And how these sexes are determined can have a variety of mechanisms. Sex determination can be genetic, the presence of a specific chromosome or chromosome balance, or it can be environmental, behavioral, or dependent on temperature. Humans have two different sexes, and the sexes are determined by the presence of a specific chromosome. Humans have 46 chromosomes altogether, 44 autosomes, those are all the other chromosomes that aren't the sex chromosomes, and either an X or a Y. The males are the heterogametic sex. They have an X and a Y chromosome, whereas females are the homogametic sex. They possess two X chromosomes. The presence of the SRY gene on the Y chromosome determines whether an individual develops as a male. Genetic sex determination can also be due to the presence of a chromosome in birds. In birds, it's the opposite of what it is in humans. Sex chromosomes are designated Z and W as opposed to X and Y so that we don't get them confused. Males are the homogametic sex. Their two chromosomes are designated Z, ZZ. And females have a Z and a W. They are the heterogametic sex. Crickets and some other insects, their sex determination is based on chromosome balance. Males are XO and females are XX. In other insects, for example, like the fruit fly, males are XY, but they could also be XO, and females are XX. The ratio between the X chromosomes and the number of sets of autosomes is what determines whether or not a fly is going to be male or female. If the X to autosome ratio is 0.5, the fly becomes a male. If the X to autosome ratio is 1, the fly becomes a female. Honeybees have a different mechanism of sex determination called the haplodiploid system. In honeybees, the males are called drones and they are all haploid. They develop from unfertilized eggs. Females, the worker bees and the queen bee, are diploid. They are produced from fertilized eggs. Clownfish are protandrous hermaphrodites. That means that they switch gender based on hormonal control. So a clownfish can start out as a male and turn into a female. The American alligator and other reptiles have temperature as their determining factor of whether or not they're going to be male or female. If you're in a nest with a lot of other alligator eggs and you encounter temperatures at 33 degrees centigrade or above, you will develop into a male. If you only experience temperatures at 33 degrees centigrade or below, you become a female. Plants can be either monoecious or dioecious. Monoecious plants are the most common, prevalent on the planet. They produce both male and female gametes in a single flower, and they can self-pollinate or you can be cross-pollinated. Dioecious plants, however, have only male plants or female plants. The plants are completely separate. There are some examples: holly, asparagus, dates, mulberry, ginkgo, currant, juniper, sago, and spinach are all the examples of dioecious plants. So here's the American holly. Below here, a good question to ask yourself, is this a male or a female plant? How can you tell? The white campion has an XY chromosomes to determine that they're male, or females are XX. You can look at the flower here and see that this is definitely a female flower because the seed pod is bulging where all the seeds are going to be, and the male flower does not have that. If you look up close at the holly flower, you can see the female flower has a distinct pistil, and the male flowers are lacking that pistil, but they have pollen. There's no pollen on these anthers in the female flower. And of course, if you get pollinated and you make seeds, those seeds are going to be encased in a red fruit. So if your holly bush has berries on it, it's a female. Dosage compensation is an important issue to consider. How is the expression from the X chromosome equalized in the different sexes? There are different mechanisms to control this. Some have increased expression from the heterogametic sex. So if you have only one X chromosome, you increase expression from that X chromosome. If you have another mechanism is to decrease expression in the homogametic sex. If you have two X chromosomes, you decrease expression. There's a lot of different mechanisms for this. In human females and placental mammals, one X chromosome is randomly inactivated in all the somatic cells. Marsupial females also inactivate one X chromosome, but it's always the paternally derived X chromosome, always the chromosome that came from dad. In Drosophila, the X chromosome in males is hyperactivated. You express genes from this twice as much to equal the amount of expression that you would get in a female. And in C. elegans, you decrease the expression from the X chromosome, from both X chromosomes, to about 50%. How does mammalian X chromosome inactivation work? Well, the Lion hypothesis stated two important criteria. One, that first of all, that one X chromosome is randomly inactivated in the somatic cells of XX females, and equally important, that the choice of inactivated X chromosome is maintained through mitosis. How does the Lion hypothesis explain mosaic coat coloration? Here's a calico cat. All calico cats, turns out, are female. What about in mice? Okay, so here is a beginning mouse embryo early on. It has one allele for white fur and one allele for black fur. Essentially, this gene has been inactivated, so it no longer produces any black pigment. And every single cell in this very, very early embryo has both X chromosomes active. Later in embryogenesis and persisting into adulthood, only one X chromosome per cell is active, and the other one is turned into heterochromatin, very tightly compacted, called a Barr body. That process of compaction or creation of the Barr body is random. In some of the cells, you notice that it is the X chromosome that contained the white allele. And in other chromosomes, other nuclei, the X chromosome that contained the black allele is the one that gets turned into heterochromatin. So after this point in development, as the mouse continues to grow and finally produce fur, all of the descendants of this cell are going to produce white fur, and all of the descendants of this cell are going to produce black fur. So this mouse will have patches of black and patches of white fur all over its coat. The random X chromosome inactivation produces these patches. What's the mechanism for turning one of the X chromosomes into heterochromatin? Well, it has to do with the Xist gene that exists in a locus on the X chromosome called XIC. The Xist gene produces RNA that binds to different loci on the chromosome itself. It coats the chromosome and recruits some proteins that hypoacetylate the chromosome. Remember, acetylation is associated with active genes, so if you decrease acetylation, you're going to decrease expression and turn this whole entire chromosome into heterochromatin. This diagram illustrates that point, and one other that's very important. So you start out with two X chromosomes, you inactivate one of them, create a bar body. Once this has happened, once one of these X chromosomes has been turned into a bar body, then you maintain that choice. Maintenance occurs from embryonic development through adult life. The inactivated X chromosome is maintained as such during subsequent cell division. So you don't suddenly reactivate this chromosome. If you turn it into a bar body, all the subsequent descendants of this cell also have the same X chromosome inactivated. Here's a nice picture of a nucleus and a fluorescent image of a bar body. In this image, with the color enhancement, you can see the other X chromosome that is much more loosely organized, has the chromatin much more loosely organized, whereas the bar body is very tightly compacted. This is a different nuclei looking at a fluorescence in situ hybridization to Xist RNA, and you can see that only one X chromosome in this whole nucleus has the Xist RNA being expressed and being tightly associated with the X chromosome. This was a very interesting experiment done by Jeannie Lawrence at the University of Massachusetts Medical School just a couple of years ago. I have to set up a few facts for you. First of all, trisomies are a genetic condition that exists in humans and in other species where instead of having two copies of a chromosome in a nucleus, you have three copies in the genome. This can cause varying degrees of phenotypes. When the chromosome that is present in three copies is chromosome number 21, it's called trisomy 21 or also Down syndrome. Okay. So the APP gene is a gene on chromosome 21 that encodes a protein that's highly associated with Alzheimer's disease. The protein is called beta amyloid precursor protein, or APP. Okay. What this image is showing is RNA fish. Okay. It's showing the presence of the APP RNA that is being transcribed from three loci in cells. Okay, this is the beginning experiment. These are mouse cells with three copies of human chromosome 21 in them. Okay, at day 0, what you can see is that the APP RNA is being produced from these three copies of the chromosome. What's also you can see is that there's some Xist RNA that's associated with one of the chromosomes, and that is because the research team inserted the Xist gene onto one of the three copies of the chromosome by another mechanism that I'm not going to talk about. So one of these three chromosomes also has the Xist RNA. Okay, at the very early stages here of this experiment, when the Xist RNA is first just being produced, all three of these chromosomes are expressing APP. As the experiment continues on day 5, you can see that one of these chromosomes is still producing an enormous amount of Xist. The question is that turning this copy of chromosome 21 into heterochromatin and shutting it down? Well, when we look at the presence of the APP messenger RNA, we can see that the two chromosomes that are not heterochromatinized are producing the APP, but this one that is being compacted is producing far less APP. And if we let it go, the experiment, until 20 days, we're still expressing that Xist RNA, and that chromosome is no longer active. It's no longer expressing the APP gene. There are several different conditions, human conditions, that are consequences of having aneuploid, which means the wrong number of X chromosomes in an individual. Okay, Klinefelter syndrome, Turner syndrome, or triple X syndrome. Klinefelter syndrome, individuals have two copies of the X chromosome and a Y. Turner syndrome has only one copy of the X chromosome. And triple X syndrome, individuals have three copies of the X chromosome. It seems as though there's a mechanism to count the number of X chromosomes in a cell. Nobody's sure of how that mechanism works just yet, but we know that it exists because individuals with Klinefelter syndrome have two X chromosomes and only one of them is heterochromatinized into a bar body. It produces one bar body. In Turner syndrome, where only one X chromosome exists in every single cell, they don't make any bar bodies. And in triple X syndrome, there are two bar bodies present in every cell. So there is some mechanism to count the number of X chromosomes, and that awaits further research. There are properties of the X and Y chromosome that make them match up during meiosis, and other properties, of course, that maintain them as separate and different distinct chromosomes. Genes that are unique to the X or Y chromosome are called sex-linked. They can be the X-linked or holandric on the Y chromosome. And pseudo-autosomal genes are present on both the X and the Y. Their inheritance patterns are similar to autosomal genes. So this MYC gene right here, which is present on both the X and the Y chromosome, is a pseudo-autosomal gene. There are also regions of the X and Y chromosome, these yellow and green regions indicated here, that do not contain genes, but however they share homology so that they can pair up during meiosis, so that when you separate the two cells, one cell gets an X chromosome and the other one gets a Y chromosome. Thomas Hunt Morgan was the first person to use Drosophila melanogaster as a experimental system for genetics. He found the white-eyed fly that we use so much in genetic research. He also discovered the inheritance patterns of sex-linked genes, because it turned out that the first mutation that he decided to use, this white-eyed fly, that gene is sex-linked. If you want to learn some more about Thomas Hunt Morgan and the legacy he left, you could go to Columbia, because that's where he was a professor, their website, and search for Morgan. This is what Morgan did with his white-eyed fly. He took the male, it was the white-eyed male that he had found, and made him to a red-eyed female that was true breeding. In the F1 generation, all of the flies, both male and female, were red-eyed, and that was pretty much expected. When he crossed these flies together, however, he got a somewhat unexpected result. Instead of a regular 3-to-1 ratio, he found all of the females were still red-eyed, but half of the males were white-eyed. So he decided to do a test cross. He took a white-eyed male and mated it to a female from the F1 generation. And this is what he got after that, a 1:1 to 1:1 ratio, indicating that this female was heterozygous for the white-eyed allele. Okay, let's do a different cross. Let's cross an F1 male that's X, has an X chromosome with a wild type allele, and a female that is heterozygous for the white-eyed gene. When we draw a Punnett square with sex-linked genes, we use a special notation. For Drosophila, we have an X chromosome and another letter indicating which gene that we're following on the X chromosome. So we're looking at the white-eyed allele, and we use a W for that gene designation. We also use a plus sign to indicate that this is wild type. When we write the male gametes at the top here, the male gametes can have either an X chromosome or a Y. Now, these genes are not on the Y chromosome, so we don't indicate that they are. When a male has a gene on the X chromosome that we're following, we say that the male is hemizygous. It has only one copy of that gene. The females, however, have two X chromosomes. Their eggs can have either the wild type allele or they can have the recessive white allele. Right here. Okay, so when we fill out the Punnett square, we're going to use the same annotation with the X and the W for the white-eyed gene. This red-eyed female got a wild type allele from dad and a wild type allele from mom. She is homozygous and has red eyes. This female, however, got a wild type allele from dad, but the recessive white-eyed allele from mom. And so she still has red eyes, but she is heterozygous. This male is red-eyed because the X chromosome that he got from mom was the red-eyed allele. This male, however, is white-eyed. So he obtained the X chromosome with the white allele, the recessive allele, and that's the only one he has, so he will express that gene and have white eyes. Duchenne's muscular dystrophy is another sex-linked gene. It's present in both dogs and humans. We can do some reciprocal crosses to see how this gene behaves when one parent has it or the other. If you set up a cross where the female is homozygous for the normal gene, indicated by a capital letter D in this case, and a male that is carrying the Duchenne's muscular dystrophy recessive allele, you can fill out the Punnett square down below here. The X chromosome in the sperm is going to have the recessive allele, and the other half of the sperm is going to have a Y allele. Mom is only going to produce one type of egg, so we can really just look at these two squares up at the top here. If mom is homozygous for the wild type allele, all the daughters are going to inherit that one allele from mom and the recessive allele from dad. They will be heterozygous, they will be unaffected, but they will be carriers. In this type of cross, all of the sons are going to inherit the normal wild type allele from mom and they will be unaffected. What if we switch the parents that carry the trait? In this case, mom is producing eggs only with the recessive Duchenne's allele, and dad has the wild type X chromosome. In this case, all of the daughters are still going to be unaffected, and they are still going to be characters, and they will have the same genotype with respect to the Duchenne's gene as they did in this case. It's just that here they inherited the wild type allele from dad, whereas over here they inherited the wild type allele from mom. Sons, however, are going to be affected with muscular dystrophy because mom has only eggs with the recessive allele in it. And if they get fertilized by a Y chromosome, then the only X chromosome available is the one with the Duchenne's recessive gene. So all of these sons will end up showing this trait. Now, what normally happens in human populations is that an unaffected carrier mates with a wild-type normal male. You should try and fill out the Punnett square to see what the outcome of that cross would be. We can infer sex linkage inheritance through pedigree analysis. Here's a population of several families that show Duchenne's muscular dystrophy in some of the members of the clan. First thing you need to look at in this pedigree is the fact that all of the individuals that are affected, all of these totally filled in symbols here, all the individuals that are affected are male. This individual, this one, and this one, they're all males. What we can infer then is that the female parent of all the males that are affected probably is heterozygous. And if we indicate that on the pedigree, then we can follow through and see that indeed all of these individuals, all of these females that are presumed heterozygous will end up having a male offspring with the trait. Notice that this female has two sons, only one of which has the trait. The other male son is unaffected. So in summary, sexual dimorphism can be determined by many different mechanisms, including the presence or absence of a chromosome, chromosome balance, temperature, or even behaviors. In some organisms, the heterogametic sex is male, and in others, it's female. Plants can be monoecious, where a single plant produces both types of gametes, or dioecious, having separate plants that produce either male or female gametes. Dosage compensation regulates gene expression from the homogametic chromosome so that the amounts are equivalent in both sexes. In Drosophila, males hyperexpress genes on the X chromosome. In mammals, one X chromosome is inactivated and becomes a bar body. The Xist gene is responsible for X inactivation by a mechanism that involves its RNA binding to the chromosome and modifying histones to compact the chromatin. Male and female sex chromosomes have some similar regions that allow the chromosomes to pair during meiosis. Sex-linked genes can be either X-linked or holandric on the Y chromosome. Pseudoautosomal genes are present on both the X and Y chromosome. Sex linkage has a special annotation in a Punnett square, and sex linkage can be inferred by pedigree analysis.